And when I became a postdoc, I wanted to do this. I wanted to make replacement beta cells throughout the body by just changing other cell types, because we have a lot of disposable cells. Muscles will regenerate, skin, your blood vessel, your fat. Those are all things that we have plenty of and we'll replace if we lose a few, right? But as soon as I mentioned to my lab at that time at UCSF as a postdoc, I was laughed at right away. My advisor said, that's not possible. And he has good reason, because beta cells are not are just as far as you can get in terms of how related they are to all these other tissues. These beta cells come from the pancreas, which is related to the liver, related to your gut, your intestine. So they're all considered endoderm cells. So what I wanted to do was to do this to convert them into endoderm cells, which are highly unrelated cells. And I wanted to do this in the body, whereas everybody's doing this in culture using stem cells. So it's a crazy idea that people didn't think would be possible. I managed to get some funds from the NIH through the New Innovator Award, where what they fund is high risk awards, a high risk, high reward award, where if it works, it would be pretty cool. And let me tell you why I was laughed at, right? Because this is what us developmental biologists or in biology grew up with. We are taught that stem cells in our body, when we start out as a single cell, and we turn into many different cell types, including fat, muscle, gut, they become very distinct. They come from very distinct lineages. And these cells are thought to be basically fixed and restricted. They're unable to become anything else. And if you can change them, you can only change them to something similar, which other people have shown. But nobody's ever been able to show them change in such a drastic way. And this is what we wanted to do. We want to change it in a drastic way and do this without even going using stem cell technology, because when you change the cell to stem cells, especially if it's in the body and even in a plate, nobody wants to put these cells back in the body because they can become tumorigenic, right? And so we want to do this directly in the body. And so we chose the zebrafish as a model system. And if you look at this image of the inside of a zebrafish, you see very similar organs. You see the heart here, the vasculature through the lungs, which is basically the gills, which is like the lungs, liver, pancreas, gallbladder, and so on. So it's very similar. Now, if we take a closer look into the liver and pancreas system, if you look at your body or a mouse's body, the liver, gallbladder, pancreas, duodenum, they're all connected by this very common, it's called common bile duct. That's the topology. And if you look in mouse, it's the same. Now, if you look in zebrafish, it's exactly the same. So they're very similar. So all I'm trying to convince you here is that the fish are just perfectly good models. They don't just look the same. Genetically, they're the same. They're not just our lab. Many other labs are showing that the fish has all the same genes doing the same thing. You look a little later, you can see here a little bit more obviously in this purple pink color here, those are the beta cells in the pancreas. So we can see this really well in the broader context. So they have very similar organ system, especially beta cell, which is what my point here. But let's move to another tissue. So you guys probably had lunch today. Hopefully some of you know what this tissue is. This is basically a magnification of salmon fillet. And this tissue is really my challenge right now, right? Which if you look closely here, you can see these are muscle fibers. So the fillet of the fish is basically you're eating the fish muscle. And each of these strands here, these are the muscle fibers, right? So my challenge for my lab was to convert these muscle cells, which are as different as they can get in terms of how related they are to the gut. And we want to make gut cells because gut cells are where the pancreas comes from, is where beta cells come from. If we can get to gut, we should be able to get to beta cells. All right. Now, what makes the challenging more difficult is that we want to do this directly in the body and without stem cell technology, as I mentioned. All right. Now, if you look in the zebrafish, we can actually turn on genes in specific fibers of this fillet of the part of the muscle part of the fish. You can see the individual fibers here, and here we overexpress. We turn on this gene called mTERI, which basically makes it glow in red, so we know which cell has our genes activated. And so how do we do that? Basically, we just take DNA, special DNA that we have, and that DNA can be have coding for genes that we're interested in, genes that we're testing the different cocktail genes to make the gut or pancreatic beta cells. And we take that DNA, we inject it into a single cell fish embryo, and it doesn't get turned on because there's a special DNA that will only get turned on if we heat shock these animals, right? And we put them in a special animal where you can see it glows in green where the gut is. So this is the mouth of this embryo, and this is the anus. You can see the whole part light up here. And normally you don't see it in the fillet part of the fish, which is the muscle part, which is right here, right? Now, if we heat shock it to induce those genes, now we can see the activation of this gut gene in the muscle. Here, the muscle we labeled in green, so we can see this patch of fillet of fish right here. Those muscle cells are starting to become gut-like. And the genes that we found, again, it doesn't matter which gene it is, but I just want to show you that we discovered a couple of different genes. This cocktail was able to do this. And I can keep showing you more data that it becomes gut cells, but basically I'll just show you one here where it becomes, it's not only does it have that gut gene come on, you can see this other gut gene come on, and you can see in blue here, that's the myosin, that makes liver. I mean, not liver, I'm sorry, that makes muscle, and that is gone. So we're losing muscle, we're becoming gut-like cell. The problem is, I can show, like I said, show this over and over, but people don't quite get that these cells are changing. And so the best way I found is to show it by using a movie. And so this is a real live animal. You can see this is the gut tissue right here. This is the tail end of the gut. That's the intestine, which is in green here. And this is the fillet part of the fish, right? And we can overexpress these two genes. And you can see they already have those genes in red here. You can just keep your eyes on those. I'm going to turn on the movie. And you can see these cells start to just ball up and change shape. And you can see they turn green. They turn green here. So they're starting to become gut cell. And they're moving in that direction, becoming gut cells. So here you can see within hours, these cells are changing so rapidly. We were actually surprised ourselves because we only saw it in different time points. But now with a movie, we knew this was happening fast. These cells change shape. They start to become more gut-like cells. And so these cells can go on. So let me show you the efficiency here first. We were getting about 30% efficiency. So keep in mind, when people first started making stem cells, the efficiency was under 0.1% efficiency. We're at 30% to start with. So it's pretty efficient in making gut cells. And it's very specific. We're not turning on these other cell types like vasculature or neural. We're making gut specifically. So this is all, that's the point of this slide. Right. So what about beta cells? What about pancreatic cells? Here, this is a gene called PTF1A. And even in human cells, this gene gets activated in pancreatic cells. And so that's how what we use to label. See, some of these cells become pancreatic cells. And as you can see, pancreatic cells do come up, they change shape. They even start to come together to form mini organs, we think. And so they move on. If we then take these cells individually and just sequence every single cell to look at their identity, we're finding, so this is the muscle cluster here, which is represented here. You can see this gene called pancreatic factor, and then insulin, which is produced by beta cell. They start to pop up too. But it's still pretty inefficient. But what we're able to do now is we can say that these cells look like they're capable of becoming beta cells. All right. And for other people who are familiar with the field, this slide basically is just to say that we can reprogram without going through a pluripotent or like a stem cell approach. But I can go back to that for some if there's more questions. So here, what we did was we then applied this technology of these two factors, and we, instead of using fish cells, now we took mammalian or mouse cells, and it looks like it's working also. So it looks like our approach to make these gut cells that we discovered in fish also applies in mammals, which means it's likely to also work in humans. Right? And so this work is under review right now at Nature Regenerative Medicine. And hopefully it comes out, and I think people will start to appreciate that we can reprogram cells in an extreme way.
Great. Thank you so much, Dr. Dong. So our first question is, how soon will this be available for T1D patients?
I mean, there's my timeline, and there's the medical and scientific field, which I think in general in a lot of fields is people tend to be what do you call it, resistant to change. Because if you say regenerative medicine, I think 99% of the people out there will say stem cell technology. You have to use stem cells. And I can tell you, when I show this, even when I show the movies, I said, we're doing this without stem cells, and we're doing it directly in the body. I would still get questions from people, are these stem cells? Are these in culture? Because they don't believe these cells can change so fast in the body. But my point is, getting people to adopt this new strategy is going to be difficult. So that would slow things down. I always use the analogy of the horseless carriage, right? Because when you first, when the first cars were invented, people always said, there's no way it could be better than a horse. A horse has all these advantages. Cars need gasoline, need all these other things. But at the end, you find out that there are a lot of advantages to the horseless carriage. And so unfortunately, I think it's going to be tough for this new approach to be adopted as a new regenerative medicine approach. But I think people will eventually come over because stem cell technology has been around for a long time, and now it kind of morphed into using stem cells to model disease, which is a great, amazing thing that we can do. But to actually take the stem cells, change it to something, and put it back into a person, that there's only very few examples of that being actually used in clinical science. In this case, we're saying that we should actually go directly in the body and changing those cells, and that there's a lot of resistance there. But we can make these arguments that, well, you change it there, the cells will have better survival. You change it there, you don't. You change it directly, you don't have the cancer risk, and so on. And so it's just just more and more people have to work on it for it to be accepted more broadly. So I'm sorry to say it probably will take a little bit of time for people to accept this approach, but this is probably reality.
Great. We do have a few questions from the audience today. So I'll go there next. Is your research directed at type 1 diabetes in the honeymoon phase?
I think it's for any phase. I think any phase where you don't have enough beta cells. And the ideal thing is for us to make beta cells. I use muscle as an example. And in fact, people have actually transplanted beta cells into the muscle, and it does work there. That's happened in people, in humans, and in mouse models multiple times where they transplant it into that environment. It works. So muscle is one tissue that we would aim at reprogramming, but we were thinking of reprogramming other cell types, including your blood vessels and so on. But the idea is to replace cells that are lost through type 1 diabetes, the beta cells. And so I imagine it would be useful at any stage.
Okay, great. Will there be a kill switch embedded if the cell conversion runs out of control?
So I love these type of questions because that means you're on board. That means you're like, okay, let's figure out how we're going to make this happen. I always like that type of question because it's more about how to make it work than why it won't work. Yes, and definitely a kill switch would be ideal, because then you can, if something goes wrong, you can turn on a gene to kill those cells. And similarly, I think there's another question that I saw earlier, and I've had this question before. What about type 1 diabetes, where the immune system is destroying these cells? You can potentially express genes that will shield these cells from being seen by the immune system, so you can evade the immune response. So that's another way. Or the other way is, if you're so efficient at doing this, you can always keep ahead of the immune system. So create some, they get killed off, create some more to stay on top of it. But I think over time, that will allow these cells to become healthy again, and if and maybe they will not be attacked anymore. Or another approach is there's a whole field of type 1 diabetes immunology where in my opinion, they have worked out their side already. It's just the developmental biologists who are making these cells are catching up, because I think there's a lot of nice data on their side where they can stop the immune system from killing these cells, but somehow we have to replace them, because even if you stop killing these cells, by the immune system. As the new ones come up, they get killed quickly because they get stressed out right away. When the cells get stressed out, the immune system can sense that, it gets rid of it, right? And so if we can have enough where they don't get stressed out because we produce so much, maybe the immune system will stop attacking it. Those things are just unknowns you have to do to find out. And it's always better to do and find out than to pretend that or come up with reasons why things won't work. And that's why I get back to my point here is I love those type of questions, because those type of questions tells me that people want to make things work, not tell people why things won't work.
Great, thank you. Have you been able to demonstrate glucose responsive insulin production from the reprogrammed cells?
No, right now we are just barely starting to be able to even see some beta cells. That's why we kind of stepped back and said, can we make this more efficient? Once we get enough of those beta cells, then we can start asking whether they will work or not. We have all strategies lined up for us to test to see if they work or not. So those are all on the way. I mean, I don't like to complain about this, but that's really the issue, is that we probably would be there already. It's just, it takes time, it takes money, it takes people to do this work. And for example, the ideas I have for making, to just even reprogramming was with me even when I was a postdoc, but it took me this time to get enough money just to even get it started. I already have the plans to how to make beta cells 6-7 years ago. I just have to build up the funds to be able to do the work, to show that we can make functional beta cells. In the meantime, we're doing other things that takes a little bit less funds. We can make neurons, we can make all these other things, but to go all the way down that path, it's going to take a lot more resources.
If this is successful, does it require immunosuppression?
Yeah, so I kind of got into that earlier. Immunosuppression, well, it depends on how efficient it is. As I mentioned, if we can stay ahead of the immune system, we can produce more before it gets killed off. That's one way to do it. We can make it evade the immune system by expressing something else to shield it. And there's a lot of technology out there that, that suggests that we can do that. Alberto just mentioned a paper earlier when we spoke. If you overexpress FGF1, that might be able to do it. So that's something that we can try. But the point is now we have this platform that we can test all these different ways of evading the immune system.
And we had a couple of questions about this specifically with children. Would it work differently? Is it similar? Can you comment on that, please?
Well, in children, I would imagine it may be a little easier to reprogram their cells, although, again, we have to do it to try, because younger cells tend to be generally believed to be more plastic, so we might be able to reprogram better in younger people.
Okay, great. And can you comment a little bit about the limitations of regenerative medicine?
Limitations. Well, there's kind of going back to my other response. There will always be limitations. The question really is more, how do we overcome those limitations? So one of the limitations was brought up. Will it be attacked by the immune system? So there may be some possible solutions there. Another limitation right now, which I still don't see as a limitation, is how do we deliver these factors, these cocktails of genes to turn on to make beta cells. There's already many labs out there that work on gene delivery, whether they're using nanoneedles, which is what I'm really eyeing, is they have these tiny needles that can actually poke individual cells where you can deliver these genes. So that's one way. There are different ways to delivering genes. There are potentially even drugs that can activate these genes. So the delivery will be, I don't want to say hurdle, but it would just be another issue, another challenge that we have to get through. But I'm going to depend on the other scientists who are working on gene delivery for that. I mean, we have to find what, another thing is, what is the perfect cocktail for making beta cell? I mean, we've gotten probably the first part of the set of cocktails. One of the subsequent ones, I told you 6 years ago, I already knew what cocktail I want to try. It's just, we just need to get the resources to start trying those cocktails. we have very we have I have very novel cocktails that I don't think anybody has tried, because I mentioned kind of hints at that without actually revealing what those cocktails are. I said synthetic and zoonotic genes. So most people have tested, a lot of people who are in this field have tested normal genes. I'm testing also newly created genes or modified genes. And being inspired by other animals. So people, it's unheard of for people to think, oh, why would we put an animal's gene in our body? But we do that all the time in science. We do that in crops. We use genes that work, we make it happen. And the way I envision this also is that I don't want to change anybody's genome. I don't want to do gene therapy. We're just turning on certain genes and it will be off after that. So we're not really changing anyone permanently, which is what I like about this technology. Actually, what works about this technology is it's transient, it's temporary. We turn on and it's off. It's not going to be different anymore. So we wouldn't even be doing what's called, I guess it's like GMO. There's no gene therapy here. That we're just adding and turning on genes and letting it disappear. So those are some of the obstacles. There are always going to be obstacles, but these to me are, how we get through it. I think that's we'll get it to work.
Great. Would this help in optic nerve atrophy?
Well, yeah. Well, to me, it's if you're referring to that as a secondary effect of diabetes, if you take care of the diabetes and the high sugar side, that should resolve itself. But if the damage is more permanent, then you would use more eye regenerative therapy, which a lot of people do work on, and our technology would apply to this. And actually, there is some studies out there that are already looking at how zebrafish actually regenerate their retinal cells, and how that inspired new technology to help adult mice regenerate their retinal cells. I know you're talking about optic nerves, but it's the same kind of concept. Is understanding how you can create these new cells or regenerate these cells. Our approach is to create them from another cell type. In that case, you need it there. So somehow you have to create, if there are existing optic nerve stem cells, you might be able to stimulate those to regrow or fix the damaged ones. So there are many different approaches. I think we just have to work on them to figure it out.
Okay, great. Thank you so much, Dr. Dong. There's only a few more questions left. What about studying T1D patients who only have positive antibodies, not yet progressed to decreased insulin production?
Yeah, to me, I think personally, I think the best approach there is to take care of the immune side, where you can stop the immune system from killing new ones. I think the combination, there is a theory out there where type 1 diabetes, which is kind of an autoimmune issue, is actually it's thought that when the beta cells are challenged and they get sick, the immune system wants to kind of take care of those cells or get rid of those cells, and that starts the problem. And so to me, it's keeping those cells healthy and keeping the immune system from attacking it. That would be the first approach. And then if you can produce new beta cells, then you can compensate for the lack of function from those little few existing endogenous cells left. So potentially, I think, we're just creating new cells, and I think it will apply in any of these instances.
How did the Diabetes Research Connection help you and your lab initiate this project?
So, of course, we had the project going already, but having extra support allows us to do a few more experiments, test a few more ideas. And this is really important. This is why we use the model that we use, the zebrafish, because we can think of like any idea, we can go test it within a few days, within a few weeks. Whereas if you want to do this with a larger animal like the mouse, it will take a lot more resources. So with funds from DRC, we were able to test a few more cocktails, run some extra experiments. And sometimes, this is what I always tell my students, if you have 20 good ideas, right, you only need one of them to work. So if you do all 20 and one of them work, it's great. That's basically what we did in the lab. We thought that there's no way we could reprogram cells. Actually, when we first started, we thought we were in the same mindset as other people. We got to use stem cell technology. So we actually loaded up on stem cell reagents and everything. And I told my students, why don't you try this? Overexpress this gene and then this gene. And it ended up working for us. And then we improved on the technology and it got better. But being able to just have the funds to ask and try these simple experiments sometimes will lead to something much greater. And if you don't try, we would not have made it here. And actually, when we did try and it worked, and our students were they didn't believe it. Actually, it took me months to get them to get back to the project. I told them, because there's no way you can change this. And they saw it. And I said, this doesn't happen on accident, guys. You got to, get with it. And actually, I lost two graduate students because they didn't think this kind of thing would work. And I managed to fool one into doing it, and doing really well. He actually has a faculty position right now from a Ph.D. program, which is pretty unusual. But sometimes you just have to try crazy things, and of course everything requires resources, and DRC has been instrumental for that.
Did the funding that DRC provided, did it precipitate additional funding from like the Keck Foundation and NIH?
Yes, so we got the Keck award after we got the DRC. And so the Keck award was very similar to the New Innovator Award, where they saw our data from the New Innovator Award, how we were starting to be able to reprogram cells. In the Keck Award, based on that work, we got the Keck Award, which is another $1 million for advancing this technology. And from that work, we introduced two new ways of making this better. So I use the analogy of reprogramming the software and reprogramming the hardware. And that came from this Keck Award, which again, because we were able to do more studies from DRC that supported that grant proposal. We're waiting for this new paper to come out, and then we'll try to get NIH funding after that.
Great. Okay, our final question today, before I bring our co-founders back, is why was the zebrafish chosen over other fish for your research?
So in a way, it was kind of chosen for us because let's take it back to let's go more philosophical on this, because I think it's an important issue. If you take mice away from biomedical research, we'll probably be 20% of what we know right now. If you take flies, which people have been studying using fruit flies for genetics for, like I mentioned, over 100 years, we'd probably be at 2% of where we are right now. If you take away yeast, bacteria, viruses from our understanding of DNA and biology, we'd probably be less than 0.01% of what we know right now. So these basic animals, these we call them simple animals, has been important for accelerating science. That's how I see it. It just makes it go way faster. You look at, more than half of the Nobel Prizes are from, study using these weird animals. Like the Nobel Prize for autophagy was won 2016. That was from yeast work. People are studying yeast. They found autophagy, which is a recycling process. And then now we're realizing, wow, recycling is so important, just like in the real world. But in cells, it's fine. It's turning out to be as important as for cancer. Now we're finding it's important for reprogramming. But in terms of fish, people have already started working on this specific fish. Mostly the reason why it's popular is it's really easy to play with their genes. We can just, like I showed you, we just pump in some DNA, turn it on, and voila, you know what happens with those genes. And you can't do that with mice very easily. In fish, you can do that. Just, you can just go over so many things. And as you saw live imaging, you saw a cell reprogram right in front of your eyes. And to me, that's, why I do science. You can see this happening. And you start with DNA, which that DNA technology came from our using viruses and bacteria. We know how to play with DNA. We take that technology, we applied it into fish. We put DNA into fish. We got reprogrammed cells. And so the reason why we use this fish They produce a lot of offspring. They're transparent. The genome's been sequenced, so we know how to play with the genes much more easily. People do study with other fish, but these fish are just more convenient. They're small. This is how the analogy I use. A whole lab can study different genes using fruit flies, and those fruit flies will fit in your closet. And for the same amount, it will take a whole room of zebrafish. For the same amount, for mouse, it will take a whole building. So that's the kind of the scale that you have. So we have one fish room. We study all these different genes, and that allows us to understand genes a lot better. If you use mice, which is very important because they're mammals and everything, you just can't do as many experiments. And with doing crazy stuff like what we do, we need to be able to do as many experiments as we can think of.
Okay, we have a last minute question that just came in. I promise this time it's the last one. I'm going to bring David and Alberto back in so they can comment after you finish answering this. But assuming all goes well, how would you envision treatment for a type 1 diabetic? Would you create something in vitro and then inject in the pancreas? And then what? That's
the question. I always get that question. Even though I said, this is all directly in the animal. We're not doing anything outside the body. Do you see what I mean? We are so ingrained with how we think about regenerative medicine. We think stem cells, we think something outside the body. We're actually doing it directly in the body. And so my envision, actually, when this technology first came out, I thought of a patch, right? Can we put a patch on your skin, just like, any patch, a medical patch, and those patches will deliver these factors and change some of our skin cells to beta cells. So that's one way, right? I thought of another way. So I told you, my mind's always crazy thinking about all these things. Another way is we can inject these factors. It can go through our whole body. It won't do anything. But let's say, so one of the ways you can get genes into cells is through what we call electroporation. A little electrical impulse will allow these genes to go, it pokes holes in the cell, these genes will go in. So I imagine we can just inject it into a person, nothing will happen. But in different parts of your body, wherever you want your blood vessels to turn into beta cells, you would then put a little electrical pulse, those genes get delivered, you convert those cells. So that's my crazy vision of how this is gonna work. The patch may be through nanoneedles. And so there's so many different ways to deliver genes these days that we just have to try them. Okay, so let me put that in the perspective of a patient. So if you have diabetes, you would apply those technology and those cells would be made, and then those cells will work because they'll produce the insulin, and then you won't need your normal insulin. And what my hope is that when you do give your normal beta cells in your pancreas a chance to relax and recover with less stress, then the endogenous, your normal ones will start working better again. That's my goal. The flip side of that is we can always just keep ahead of the system, the problem, always making more beta cells throughout your body. We have lots of vasculature, we have lots of muscle cells, plenty of fat, plenty of skin. So that's my vision of how this will happen.
Great. Thank you so much, Dr. Dong. Before we conclude, which David will do for us, I wanted to give Dr. Hayek a chance to respond or comment on your presentation.
Your mute's still on.
Okay, you're unmuted now.
Okay. Nice presentation, Duc. Just a couple of questions. I guess you are waiting for your muscle cells to show the insulin protein before you move into a bigger animal. And I was thinking when you were talking about how to take these cells from definitive endoderm to an insulin producing cell, I remember a paper many years ago by Doug Melton, where he took an injection of adenoviruses containing 3 transcription factors, like NeuroD,
Neurogenin 3,
PDF,
MAF-A, and PDF.
Exactly, okay. And he just injected into diabetic animals and he cured the diabetes. But apparently it didn't work in vitro, which gives a lot of validity to what you are doing in vivo. Is how complicated it is to inject into the muscle-adjusted package with these 3 genes to see if they go from definitive endoderm into a beta cell.
We've actually done it.
What?
So we have very preliminary data. We actually injected our factors into the muscle of mouse already. I had to halt the project because we ran out of funds, but we did see some sign of a PDX coming up, just using R factor, which has no PDX. So it does look like it is likely to work. Like I said, we ran out of funds because the mouse experiments are a lot more expensive. We went back and said, well, we know it's kind of working. Let's see if we can advance this technology, optimize it in fish, and then go back to mouse again as soon as we get more funds. So on our side, we're trying to optimize it in terms of making it more efficient, but also change better into a beta cell. So like I said, we have factors that I've been wanting to test beyond the factors that I showed today. But yes, we have tried it in mouse muscles, and it does look like it's working.
Going back into this idea of once it works, making insulin, do you have a second cell line or organ that you would like to put this treatment to regenerate? Actually, what I'm talking is endothelial cells. Could they be targets for your therapy? Because one of the biggest problems, even if your method works, which will be wonderful, still is peripheral delivery of insulin. And the goal for a perfect treatment will be direct delivery of insulin to the liver. So can you see one day picking up different systems such as endothelial cells?
Oh yeah, definitely. That was actually part of our original proposal. We want to hit all these other cell types. Vasculature, as you mentioned, the blood vessels are my ideal tissue because they're everywhere, and you constantly destroy your vasculature all the time. You hit yourself, you get a little bruise, they regenerate, they're fine. But what Alberto is mentioning is the blood vessels, so there's already a blood supply. But that's actually also true with any other cell type that you have in your body. All your cell types in the body has to have some kind of nutrient supply. But blood vessels I like a lot because not just for beta cells, but blood vessels tend to be associated with stem cell populations in your body. So I do want to make beta cells, but I want to make neural stem cells, so like dopaminergic cells for Parkinson's. I want to make, that's why I showed you we're starting to make neurons now. So the vasculature gives you access to the whole body because the blood vessels throughout your whole body. So if you can access the vasculature, you can access the whole body. So that's really what we want to do. We want to be able to change those cells. The main reason why we started with muscle is not just because people transplanted beta cells into muscle. Muscles are really easy to spot. You look at them, they're elongated. You know that they're mature, if they have multiple nuclei, they're striated. When they change, they change. Whereas all these other cell types, there's so much shape. Here, muscle, we knew that when they get reprogrammed, we know how it looks like when it gets reprogrammed. But yes, for sure, my ideal is to be able to change anything into anything, but I want to target specifically blood vessels because that will give me access to anywhere in the body.
Okay,
even
my last question. So you have the muscle from the zebrafish making beta cells. What do you think the membrane of the cell muscle would look like? Cell muscle or a beta cell? The cell membrane.
Oh, I think they look like beta cells. So everything we've seen so far looks like beta cells because the membrane of the cell is comprised of proteins that are coded by the nucleus of that cell. So whatever that nucleus, if we can change that software, then we will be producing the things from the beta cell itself. But what about old components? I mentioned autophagy, the recycling. So we have evidence that all sorts of parts of the cells get recycled. So we have basically a blank slate where we can just refill it with beta cell hardware. There's one issue that I did not talk about, which I want to go into the future. So I mentioned the two things that we need to change: hardware and software. We actually think we are also starting to change the microenvironment of the cell. We have some data there, but I don't want to go into that yet. It's still very early. But keep in mind, our gut cells themselves are surrounded by muscle, right? When I first started, it's like, how can we get beta cells, these gut cells, in the middle of the muscle? How can they exist there? And then you think about it. Your gut moves the food in your body because there are muscles squeezing that. And so gut cells are normally surrounded by muscles. So they feel completely comfortable in the muscle environment. That's probably why when people inject beta cells into the muscle of mouse or humans, those beta cells work. So I think the environment is actually okay.
Actually, you know, that will really be key if you could turn the muscle cells in the gut into insulin producing cells, then you have almost direct delivery to liver.
Yes, yes. I mean, that's our dream, is just, convert muscle vasculature.
Listen, beautiful work, and congratulations.
Thank you.
Wish you success.
Thank you. I really want to push this further. It's just, I feel like I'm very appreciative of DRC because I really wish people would adopt the DRC model more, because the way funding works in traditional science, it's just way too slow. I mean, I feel like I'm going at 10% capacity of what I want to do. The projects I'm doing now are things that I thought of 10 years ago. I should be working on projects that I'm thinking of right now. But that's the way, the bureaucracy is. So, not to end it in a sour note, but that's unfortunately, I wish, I guess discovery was facilitated a lot better. And there's just too many naysayers out there. People keep saying things don't work. I think it's all about how to make things work, not how things can't work.
Thank you so much. David, if you can please give us your closing remarks, and then we will close out at 3.
Duc, I think you ended on a positive note, because what you're saying is that there's a real need for entities like DRC, in particular, because we have a very rapid funding cycle, thanks to Dr. Hayek and establishing our 80-member scientific review committee. We make sure and do it fairly quickly that all the science is worthy of being funded, that it's novel and innovative like your science, and that it has a reasonably decent probability of success. So you, like many of our other grantees, have received considerable follow-on funding. We actually have someone that got upwards of $4.5 million in follow-on money, and several others like yourselves, a total of 5, have received at least $500,000. Many have published in very reputable journals, as you have, and hopefully you'll have another publication very soon. So we appreciate your good work, your creativity, and that you're not giving up, because type 1 diabetics like myself and others who are probably listening today are just overjoyed that you're willing to take on these risks and pursue science that you've pursued. For folks that are watching today, I hope you'll take a look at our website, where you'll see not only your project, but a number of others we've already funded and ones that are actively seeking funding. We can turn these projects around in as few as 12 weeks or so for full funding. So unlike other funders that can take 1.5 years, 2 years, we can move a lot more quickly. So we appreciate what you've done. I'm not sure after having salmon for lunch that I'll ever think of salmon quite again. But again, thank you for everything.
Thank you so much. Thank you to everyone that joined us. As one of our audience members says, thank you, Dr. Dong. You gave a very informative and hopeful talk today. So thank you for your work. As David said, thank you for taking the risks, and thank you for letting DRC be a small part of your journey.
And I really appreciate this opportunity from DRC and this opportunity to talk about the work. I'm very hopeful that this will keep moving, and hopefully in the future when I talk to you, we'll show you a functioning beta cell.